he'll be recorded. Posted on YouTube. All right.
Okay. Thanks, Martin. Okay. We're recording, but we need slides. Okay. Well, the slide that was up there is collage that I made almost every year since I started here in January of 2004, and it's probably embarrassing to some people, but certainly not me. I'm not someone who is afraid to put on a skirt for the right occasion. Yes. Okay. I'm gonna start my timer so that I don't impose too much on you. Okay. So before I get into the details of the science, I thought to. Oh, now I'm at the top. That's good. This is very exciting. To amuse myself, well, beyond this amusement that's currently going on, I've done a little historical look back at mine too. I never left here before. Right?
High self-view.
Yeah, because that's what's showing and that's what's keeping the screen small.
Where is it?
It was there.
Is this an elected position he has?
Self-view, this one?
Well, there's some really famous people on this list. Don Fred McClafferty, Catholic State, Roman Zubarev. A sheep. I probably can. Yeah, let's see. There we go. Well, maybe we should just go for it. Okay. So before I review some of the science that we did here in the last 9 years, there's really no way to review all of it. Having been here so long, I'll just focus mainly on some proteomic aspects of technology. But I thought you might be interested in how I actually came to be here and how I came to be interested in mass spectrometry. And now that you've all eaten, of course, I'm going to start out with sort of a gross talk, just a little warning. So if there's no visual grossness, but there'll be some mental grossness soon. So long ago, I started out in chemistry at Auburn University. I did a chemistry degree and a master's degree at Auburn, trying to figure out what I wanted to do with my life. I knew I was interested in biochemistry. Auburn didn't have a biochemistry department. All the biochemistry happened in the animal science department. And in the animal science department, they had a hog farm, of course, that's what you have, cow farm, hog farm, sheep farm. And they had these rather large boars. Now, this isn't one of the ones that I was familiar with in the biblical way, as you'll see in a minute. But this guy is 890 pounds. He's a monster. But the ones that I had to work with were about 600 pounds. And just keep in mind the severity of this. There was a pig farmer in Oregon who was recently eaten by his pigs. He somehow collapsed in the pen, and they just found parts of it. So I was interested in biochemistry, and at the time I was interested in calmodulin and calcium, structure of calmodulin and why it bound 4 calciums. And the biochemistry part sort of led me to the acrosome reaction, which is a reaction that happens when the sperm and the egg fuse. And that at the time, it was known that calcium and calmodulin were involved. And so I set about trying to purify a protein from porcine seminal plasma. And in order to do that, of course, you need some seminal plasma. That means I didn't need a lady in a red hat, but I did need a sow. And just to cut to the chase, you just read quickly here. This says something like disposable, and that's the key thing, disposable artificial vagina for collecting of seminal plasma fluid from animals. It's patented. Now, I actually didn't have one of these. This was in 1982 when I was doing this. All I had was, you know, those little thermoses are like this size, but big around that you used to take in grade school to, you know, Kool-Aid or milk or whatever. That's what I had. And I had gauze and a rubber band around the top. And you could sort of use your imagination, but we had to get this lovely lady or her counterpart and this not so lovely gentleman in a pen. And the pen wasn't very big. It was about the size probably of 3 automobiles. And you got in there with that uh, collection device, thermos. And at the right moment, you had to make sure that this guy was convinced that he was still involved with her and not with you. Because you wanted him to stay up on top of her, not jump off because you were down there. So, needless to say, this sort of experience got me interested in. I hate to. This is going to be an obvious pun, but it got me more interested in the hard sciences, so to speak. And, although I wasn't interested in physical chemistry, but I was really interested in entomology. And so from Auburn, I went up to NC State where they had a proper biochemistry department with no animals except the people in the department. Um, and there I fell in love with mass spectrometry. This is the mid-'80s. Fast atom bombardment and magnetic sectors were really the instruments to have at the time. And so I learned from Richard Van Bremen how to run these complex instruments and did peptide sequencing in the mid-'80s. Frank Armstrong was my protein chemistry advisor. From grad school, electrospray, of course, was invented when I was in graduate school, and Dick Smith's lab was one of the early adopters of electrospray. So I went way over to PNNL at the time in the early 90s and worked with Dick to learn electrospray. And at the time, as a biochemist, I thought electrospray was really pretty cool, and that's really why I wanted to go to Dick's lab. Whoops. And you can see we did some early, what would now be called native MS work. At the time, I remember thinking, this really isn't going to go anywhere, so I don't want to be fooling with this anymore. It was a lot of work. We did actually figure out the enthalpy of dissociation for this small complex using electrospray. But it was a lot of work, and like I said, I really wasn't convinced, although Matt Bush is smiling here in the front because he's making a whole career out of native MS. So I decided to leave. It wasn't really academics, but it was an academic kind of environment, and went to Big Pharma and went to Johnson & Johnson in New Jersey. So you can see I've already gone across the country twice now. And at Johnson & Johnson, I did something back in 92 that is really in vogue now, selected reaction monitoring. And so you might wonder why our lab doesn't do much selected reaction monitoring. So I did as much as I ever want to do back in 1992 on hundreds of clinical samples, just over and over. We didn't even really have good computer routines. We would graph things out and look at the results at the end of the week. So that was fun. I learned a lot, but as things happen in industry, often ideas come up and they go down, and when they go down, the bosses just shut the whole thing down and send you away. So thinking I was going to be laid off, I found a job in Seattle with Bristol Myers Squibb, where I was employed sequencing MHC peptides like Don Hunt was doing at the time. And from actually at Bristol Myers Squibb, after about a year, they said, well, we're going to close this site. We want you to move back to New Jersey. I said, no, thank you. And nothing against New Jersey. We just really like Seattle. And so I was fortunate enough to meet these 3 guys. First, Rudy, just by accident. I interviewed at Zymogenetics, and I didn't get the position, but Rudy was there consulting on the interview and called me up later, and I ended up taking care of Rudy's mass spectrometers at the UW, where I met Lee Hood, who then hired me to set up the proteomics facility at the Institute for Systems Biology. And when I was at the UW with Rudy, I met Sid Nelson, who was the former dean of the School of Pharmacy, who then recruited me from the ISB to come back to the UW, and that was more or less 9 years ago. So really, I owe a huge debt to these 3 guys who really, they say you need to be ready to be lucky, and I guess I was ready, but I certainly was lucky to have met these 3 guys and moved along the career. So this chart attempts to describe what we've been doing the last 9 years. Green is sort of algorithm development, although we're really sort of pragmatic algorithm developers. We're not fancy algorithm developers, but we make things that can work, may not work in the most efficient way. Sonia Ting's in the audience tonight. She wrote this program for automated lipid A structure development. There's a bunch of cross-linking software that we've written. One of these was with Bill Noble's lab, sort of like a sequenced version for cross-link analysis. And Sean McElwain, I saw earlier, he's here somewhere. He did that. And then we've also written a top-down algorithm. We've done quite a bit of just what I would call basic structure analysis, protein complexes, and lipid A structure diversity. I don't really have time to talk about this half pie chart. I'll talk a little bit about proteomics, not so much about lipidomics, but this, of course, does make a pretty big chunk of what we've been doing. And this purple piece of the pie actually is growing a bit lately. But we've done a few interesting things, put DREAMS on our Bruker quad in front of the FDMS. This is a device from PNNL that allows you to notch out a single mass to charge value within a precursor spectrum if you want to, so that you can create or increase relative abundance of things around it. Of course, ion funnels also came out of PNNL. I'll talk a little bit more about this 2DMS and SON in a minute. Okay, so soon, actually at some point next year I'll move completely to Baltimore. The family and I won't physically move until about June or July. But in Baltimore, we'll be setting up an institute that's built around mass spectrometers, although I've been told that I can't put the word mass spectrometry in the title of the institute. So if anybody has a good idea for a name of a mass spec-based institute that does small molecules and large molecules but doesn't reference the word because the dean can't explain it or doesn't want to explain it, let me know. It's not that easy. Okay, so back to proteins and protein chemistry. The guys that taught me enzymology from the seminal plasma taught me quite a few things, but the basic principle really of protein chemistry in those days when you made your own enzymes to do whatever you're going to do is that in large part 50% of some protein, you just don't know which protein that you're working with, is going to be lost due to adsorption and other mechanisms while you're doing your purification. So minimizing sample loss by preventing just simple pipetting steps or going into a test tube, Eppendorf, is paramount success in many cases where you've got low copy number proteins. So some of what we've been doing in the last 9 years involves these three sort of principles: sample preparation, ionization, and mass spectrometry. And I just want to go through each of these. This one is not very well developed. I mean, we do the standard things for proteomic analysis. You'll see we're headed down a different path here in the future. Ionization as well, we're doing some new things, and this will sort of wrap up this will really just be a review of proteomics we've done since I've been here. With a big emphasis on we. Like I said, I made one of these collages every year at Christmas, usually drinking way too much. Like 1 year, a student wouldn't give me her picture, so this is her picture. This little thing with a magnet for a head. Okay, sample handling. This is work that we started with a collaborator at the University of Edinburgh, whom I met many, many years ago, and ended up being chair of his scientific advisory board. This is known by a couple of names. One of them is digital microfluidics. The other one you might have heard of is EWAD, for electrowettable on dielectric. And it's basically a hydrophobic coating over a set of electrodes. And those electrodes, when they're fired in a certain order, allow you to move droplets around on a surface. So we've begun to work on this as a means to minimize sample handling. And this is an example, a couple of examples are coming of why you would want to do this, perhaps. Pat's lab in Edinburgh also has recently shown that you can work with magnetic beads that are derivatized with an affinity reagent on the chip. So you can pull out a protein of interest on the chip, mix it, wash it, digest it, And in their case, they pulled the sample off the chip and then went to the mass spec and showed that they could get lots of sequence coverage. This paper actually just came out this week, and Aaron Wheeler is one of the premier labs in the world doing digital microfluidics. And so to get your mind around what we're trying to do, I'd encourage you to take a look at this paper from his lab, which I don't really have time to go through, but it explains the basic principle of working with derivatized magnetic beads, and in his case, he's developed some immunoassays. You can see Abbott Diagnostics. Ionization. So also while I was working as chair of the advisory board in Scotland, I met this electrical engineer, John Cooper, who was working with these surface acoustic waves. And these waves actually can be used in the same way that you use digital microfluidics to move liquid around on a surface. But the other thing that they can be used to do is to nebulize samples. And the idea here is you've got this transducer across which you run an electrical pulse, and a piezoelectric material, in this case it's lithium niobate, and when the pulse fires, you send a ripple through the material like an earthquake travels through the surface of the earth. And at the point when that ripple hits liquid that was deposited here, the surface tension of the liquid breaks, and you basically, if it's close enough to a mass spec, the mass spec's vacuum will pull in the nebulized droplets. Now, at the time, it took me 2 years, for some reason, to convince John and Pat to try this, put it in front of a mass spec. But we finally did. Let's see if I can get this movie to work. This is just red food color here. The dogma is if you don't put a charge on your sample, you're not going to detect ions. And in this case, the sample sits on this grounded electrode. You can apply voltage. We don't always need to apply voltage to detect ions. And the transducer is here in the back. So there's a lot of papers now from our group coupling this technique to mass spectrometry, and Scott Edgar, who's in the back row, is administering as a PI on a grant from the NSF, this company that was spun out of the C4C here at the UW a couple of years ago to develop this as an ion source. So hopefully you can see, well, I'm getting ahead of myself, but one of the nice things about SON is that it's got lower energetics than electrospray. And Yue Huang, who's a shared student between my lab and Frank Turacek's lab, has done these experiments now to show that there is a higher survival of this precursor ion here for this benzylpyridinium ion. R is going to be a bunch of different groups you'll see, but we're measuring survival of this benzylpyridinium ion to gauge ion energetics. So here there's 2 panels. The top panel is just one temperature comparing SON in red and electrospray in blue, 4 different R groups, but pretty much in all cases, SON was energetically lower than electrospray. This is all done on a thermo instrument with a heated capillary, so that's what these temperatures are. Lowest temperature to highest temperature, SON was pretty much always the lower in terms of ion energetics. Biggest difference here was at about 225 degrees. Of course, just heating the thing perturbs the system and adds some energy, but still it's about a half an electron volt lower by SON electrospray. Yep.
On the surface?
That's a great question. I don't think so. Is Yue in the audience? Sungwon says no. It turns out that it doesn't matter so much. There is a little bit of a drop in the energetics if you add the voltage. It's easier to ionize if you add a voltage. So this is just a practical example. This is electrospray of some phospholipids. And this is SON of the same phospholipids. You could argue this isn't all that impressive. I mean, it's about twice the precursor ion here is by electrospray. But for some compounds, it's even more impressive. Like with retinoic acid, we get a huge signal by SON that we don't see very much of by electrospray. It's just MS. And feel free to interrupt. I think where this really has some promise, at least with protein chemistry, is with non-covalent complexes. And Lucas Monkonen is a student in the lab who's been working on this viral protein dimer A55, which is a fairly small dimer, or protein. The red charged states are from the monomer. Blue, we're not sure about, and green is, we think, dimer. So this is electrospray. It turns out if you take the exact same solution in concentration and you run it by SON, you always see a shift to lower charge state by SON. I'll come back to that in a second, but what you see immediately is this 8+ ion really jumps up off the page. So Lucas is in the process of doing some work with Matt and some work in our lab trying to actually do HD exchange on this particular protein and then sequence that exchanged protein using ECD. It's a pretty tall order because the sensitivity here is not all that great. But he's a graduate student. You're not here, are you, Lucas? I don't think so. He needs a challenge. What I was going to say about the charge state shift is we don't really know why this happens, but it happens a lot. We see this shift in charge state. The lithium niobate is a piezoelectric itself, and the physical perturbation, of course, is dislocating electrons. It could be that we've just got some charge reduction that's going here on the point of the sample, but we didn't prove that, and probably Scott Edgar is nervous in the back with me saying this, because we certainly didn't prove it. It's just me postulating, and them trying to think of an experiment to prove it or disprove it. If it actually is charge reduction based on the piezoelectric material. Okay, this is what I was about to jump to before. One thing I'm known for is not having rehearsed my lectures, so I certainly forgot. Scott Edgar and Daniel Gruber, who was from Pat's lab in Edinburgh, Daniel came over and Scott and Daniel did this combination DMF-SON HDX experiment just on angiotensin, but it proves sort of the basic idea that you could pipette something onto the chip, DMF chip, and pretty quickly carry out biochemistry or chemistry and measure it immediately with the mass spectrometer. And the reason Daniel came over is Daniel had a different way of doing DMF. It's basically screen printed on a polyimide sheet. So these electrodes that you saw before on that glass-looking component are actually just screen printed on here. So if I go to the next slide, this is admittedly a little bit of a hokey example, but we put 4 microliters of D2O and 4 of angiotensin in water and fire to pulse mixed. And now we have this 1 droplet, and we need to get that onto the SON chip. And in this case, we have no really clever way to do it. So it's basically this is hydrophobic, this is hydrophilic. So we transfer it the old-fashioned wicking. But once that's done, you can fire the SON chip and immediately see within seconds exchange of deuterium or hydrogens for deuteriums. I think that's the last one. So this is as far as UA has gotten. Because that's a little unwieldy, UA has basically figured out a way to microfabricate both the DMF platform and the SON transducer on one lithium niobate chip. So the next step really is for her to follow some simple experiments, I think, do some chemistry on chip by mixing some components and firing immediately in mass spec to do some measurements. So that's going on. I expect that, she needs to finish her PhD by the end of 2013, since I'm leaving in 2013. So I think that's going to be done hopefully by spring quarter when I need to actually physically leave. Okay, new methods for proteomics. This is a couple of topics, but the first topic sort of got us thinking about the second topic, and this is what you could call a failed experiment, but it taught us some things. This is work we did with Ola Wong and Gary Nugent at Micron, who's now part of Bruker. And Hola actually used to work at Bruker building ion traps for them. But so the idea was to fill up an ion trap with 1 chromatographic peak's worth of ions, and now scan out the ion trap 1 M/Z channel at a time into a collision cell after which those ions or precursors or fragments are read out by a top. So each one of these instruments is slightly faster than the one before it, so this in principle works. Some problems with this design are that scanning anything out of an ion trap, trying to slow it down at the time, was difficult. Thermo later solved this problem in part with their C-traps. You'll see in the next slide that we're probably over-energizing ions as they exit and certainly as we then fragment. The other problem with the device is that ion traps, of course, have some capacity in terms of the number of charges you can put in there. So putting a whole chromatographic peak's worth of material in an ion trap at one time limits the number of any of those ions at any given MO channel that you can put in there, and that will come up again when I get to the next topic. So here you can see results. We sort of simulated a chromatographic peak by infusing a digestive myoglobin, and we could blindly, using a database search, identify myoglobin from this data. What you see here is fragment ions, precursor ions. These are covered up because even though I'm an editor at RCMS, I still put daughter ions here and parent ions here when I shouldn't put daughter ions here. So I can tell you all that, but for most audiences, I just put fragment ions. So what you would see if there was no fragments is just a black diagonal line. So all of these are fragments that come over from this axis. So the nice thing about this instrument, even though it wasn't very sensitive, is that in one shot, you've got all precursors, all fragment ions, all neutral losses, all MRN transitions. I know that Hola and Carey met with some people at Thermo shortly after they filed their own IP, and I don't know what Thermo's doing, but Hola said he felt like they were pretty interested, and they certainly never called Hola and Carey again, which was too bad for them. But I think this is one place that mass spectrometers are going in the future, is to put a sample in and basically be able to profile every M over Z channel at every moment in chromatographic time. That's sort of one of the holy grails of doing complex mixture analysis. I mean, we can get all precursors easily. Getting all the fragment ions is not so easy. And that's why you've seen all of these data-independent methods like Waters' MS to the E method and many other methods like the Macasse lab was published. And we actually just wrote a review with Christophe Massillon, who wrote one of the first data-independent papers. And the review is on data-independent proteomic analysis. So I was surprised to see there's many dozens of papers out there describing different kinds of data-independent methods. So what we learned from this approach led us to try getting away from this data-dependent approach where you pick ions. And what we're doing, before I show you the next slide, some of you will have seen this before. This is a slide from Yonser and Yates's review. But normally you fill up the ion trap with all the ions from whatever, 1,000 or 300 to 1,000 or 1,300, whatever you want to fill it up with. And to look at any given ion, you've got to, if you want to look at the middle ion in yellow here, you've got to get rid of this one and this one on either side. So in the end, you've filled up the ion trap with a lot of things, and most of it you're not interested in, at least not at that moment. So you really haven't done yourself any favors in terms of the concentration of this. So a couple of Swiss guys, Alex Scherl and Alex Ponchot, when they were in the lab, developed this. I had a few laughs with Jim about acronym development, and I have a few acronyms that have faded in the dust of time. This may be another one. But the Swiss guys thought this was really funny that they could come up with this acronym since they were living on the Pacific Ocean. But it was a lot of work. Anyway, the basic idea comes from our gas phase fractionation method, where we inject the sample multiple times and do data-dependent analysis. Except now, we are going to inject the sample many times, but now we're just going to scan single channels. And for various reasons, they need to be overlapping to improve the number of hits. And early on, we could only do about 15 M over Z per LC run, and the first experiment took 5 days, actually, to get through 1,000 M over Z. And the Swiss postdocs actually weren't too concerned with that. They realized they could take the week off and come back and then start the data analysis and take another week But of course, if you're trying to get a lot of samples through a facility, then people aren't so keen. Currently, John Chapman and others in the lab have this down roughly to about 24 hours using one of the captive spray sources that Microme produces and now a group sells. But so the basic idea is iterative analysis. This iterative analysis, systematically scanning every channel, gives you a number of benefits. This is all compared in blue to gas phase fractionation, which is a data-dependent method. So the number of proteins we ID goes up, the number of peptides we ID go up, and sequence coverage goes up. Interestingly, we found 3 times as many transcription factors, but this was from a Pseudomonas aeruginosa lysate, so you can't really say too much about copy number. So Sun-Hee Jung in the lab, who's now, I think, at Children's doing MRM analysis, used a yeast sample that sort of a standard yeast sample will allow you to calculate or estimate number of copies you detected in your mass spec experiment. So in purple is a paper that I'll come back to at the end from Eric Voss. It was actually one of the first things we did when we started with the UW. This is all data-dependent analysis, and no fractionation in any of these. Well, in Matthias Mons there is, but so purple, there's no fractionation, just whole cell lysate, shoot it on the mass spec, and you can see that's centered around 30 to 60,000 copies. White is actually the tap tag numbers where they've estimated copy number. Blue, or light blue, is about 400 LCMS-MS experiments that Matthias Mons group did, and to get to that, they did gas phase fractionation, although they didn't call it that. They did isoelectric focus, and they did a one-dimensional STS page. They did a huge amount of work, 400 experiments. They do identify more proteins than our Pacific here in orange. This is 40 experiments, or 40 injections at the time, which is now about 25 injections, I think. And you can see that we're probing more or less the same detectable dynamic range as this massive experiment, and certainly as the tap tag experiment. So the other benefits of this approach is because we're systematically looking at every channel. Sun-Hee's estimated in her yeast sample that we're up to about 90% reproducibility, and we get a detectable dynamic range that looks pretty similar to what Rudy's group reported. They used a similar yeast sample and did SRM on select proteins, across this detectable dynamic range scale. So this is supposed to be the comparison to plasma, and this isn't a very clever slide. I meant to have this show up second, so just ignore this for a second. The Pacific results, we got about 500 proteins with a 0.5% false discovery rate, and there were several papers. I don't want to just pick on this one, but there were several papers that did a mud pit kind of experiment where they fractionated, and you can see here they had 144 fractions that they analyzed in duplicate, so almost 300 experiments. And from one fraction, we identified twice as many proteins. So I think you really do have to ask the question, why fractionate if you're going to take one of these, this kind of approach? We identified about 80% of the same proteins and 3 orders of magnitude better detectable dynamic range. Now, we do have to inject the sample multiple times, but I think this added detectable dynamic range when you're building up a proteomic database is an advantage. Certainly, injecting it many times is a disadvantage unless you're not sample limited. And this is a slide from Shauna Hingel's paper where they were looking at tamoxifen-treated MCF7 cells, and we could see on quantitative PCR that AGR2, which is a cell rescue protein that the cell turns on to try to thwart the killing mechanism of tamoxifen, is upregulated. On Western, we could see it was upregulated, but this lower band here is GPF. It's a data-dependent experiment, and we could detect AGR2, but it wasn't upregulated in a statistically significant way. So if we had just looked at this proteomic data, we would have never put it on the list of things to follow. So I think the point here is that we're getting more sequence coverage, which allows spectral counting to perform at a better level. And then the final thing, Young Oz here, hope she's not going to shoot me for putting something that's not published, but I think this is an obvious next step, even though I'm not crazy about SRM. This is an SRM-like experiment, but it's specific. And so really the only difference is we're acquiring all the fragment ions, not just a few fragment ions. And this, of course, uses the Makasis Lab Skyline routine. But she's basically working on this combination discovery validation workflow where once you know a few of the proteins in the sample, spike in heavy label peptides and run with that. So you have the ability to quantitate what you already know is there, but also the ability to continue to identify new proteins. Okay. All right. So now I want to go way back to actually the start of the group. Because in some ways, this simple sort of proteomic set of experiments that we did with at the time is Eric Foss and Leonid Krugliak, who subsequently moved to Princeton, and Tony Bedelov on a yeast study and leukemia study, actually has proven to be it's the simplest thing done technically, but I think in terms of the value that we've gotten out of this data, probably the highest. And of course, the problem with clinical proteomics is you want a lot of time points. You don't want to just look at time point 4 and 1 because you may miss these dips that are indicative of some sort of behavior that you're interested in. So really, to get to this approach, you want to minimize sample handling and fractionation. Unless the rate of scan speed of mass spectrometers improves by many orders of magnitude, I think you want to do what we've done, which is basically lyse the cells, digest, and put them into the mass spec to get as much information as possible in as simple as a manner as possible. So just to see if you're still awake, what you really need for good clinical proteomics is well-defined phenotypes. So, of course, this is these photos are taken about 30 years apart. So these are very well-defined phenotypes. If you have other kinds of phenotypes that are less well-defined, these are not so easy to pin down with proteomics. You'd argue that you need many more time points here than here. You could just probably get away with these two samples. The other point here is that each person, at least with human clinical samples, probably makes the best control. You certainly don't want to use other people as control. Heterogeneity is so high that it becomes very difficult to start to pull signal out. Okay, so literally from the time that actually before I left the ISB, Eric came by, and Eric was trying to do this following experiment with ICAT, which is a cysteine labeling method that was developed by Rudy before he went to the ISB, and it does involve fractionation. And so Eric had two parent yeast samples, and he wanted to ask the question, could he follow inheritance of quantitative traits? So like height, can I see how height is propagated through yeast? In this case, it's not height, but height with humans is a quantitative trait. We're trying to follow similar quantitative traits in yeast. So he wanted to know, can I follow these quantitative traits using proteomics? And he had started to do this by ICAT at the time and basically had given up. And to make a long story short, he started very early on doing label-free proteomics, mainly because you could acquire each data set from the sample without having to worry about mixing it with another sample and multiplying errors later on when you want to know, if you've done the experiment with A and B compared and you want to know something about C and D relative to A and B, you really just start to multiply errors in a bad way. So acquiring data on each sample independently has a lot of advantages for clinical analysis. So Eric had 100 yeast siblings, all from the same two parents, just from different perturbations, a lab strain of yeast and a wild vineyard strain of yeast. We just did simple total protein extracts and then quadruplicate LCMS data, time on an LTQFT. So parent ions are acquired in the FT and fragment ions in the LTQ. From this, we had about 400 data sets. This is by roughly the end of 2005. It took, after many mistakes in trying to figure out how to do this best, it took about 6 months to generate this data. And they paid us $100,000, actually. It wasn't enough, but that's how much money they had to pay. And hopefully somebody from the hutch here, am I going to get in trouble with administrative people? Nobody wants to admit it. Anyway, Krugliak was leaving the hutch and moving to Princeton, and he had this pot of money that he wanted to get rid of. He didn't want to leave it, and he couldn't take it. So you paid it to us to generate these data. Thanks, Leonid. So there's also I hope there are no Croatians in the audience. I'm a big fan of Croatians, but there was a crazy Croatian involved here, Dragan Radulovic, who's a mathematician who had figured out this way. So these are just ion maps, so you're looking down on m over z versus time here. And of course, there's this nonlinear adjustment that has to happen, and this is Dragan's mathematics that I don't understand. But it allowed this nonlinear transformation, and so now spots start to line up from different LC runs, and from this we could readily extract these. And all of this got published back in 2007. So just as an example of how the technology actually is working, here's 10 replicates of one parent, here's 10 replicates of a different parent. This is a particular peptide that's polymorphic, which means that you should only see it in one parent and not the other one, and that's pretty much, at least within the bounds of detectable range here, that's what we see. Not sure why we have one outlier there. So when siblings inherit this from their parents, right, I have two sons, one's taller than me and one's shorter than me. So the quantitative traits are sort of distributed, and as you'll see, in this case, about half of the siblings shouldn't receive this quantitative trait at all. And in fact, they don't. If you took the time to count, you'd see this is roughly 50 down here, and the other 50 have some variation in that quantitative trait that they've inherited from their parents. So, and this isn't trivial. I mean, it does depend directly on being able to manually go through the quantitative data and map things out, look at what makes sense. We all know there are peptides in these bottom-up data sets that don't make sense. So it's pretty labor-intensive in terms of the analysis. And of course, at the time, and probably still now, in large part, we all have to do these experiments for the Western blotting world just to prove that all the mass spec data lines up, or at least in this case, we had to do 9 of these to prove that it worked as well as Western blotting. This actually is an advertisement for selected reaction monitoring. So the final study also involves Eric and Tony. Leonid's moved on now, but Tony's a physician at the Hutch. And so we moved on to do the same thing with some leukemia samples from patients that Tony sees. And so the basic question is can we distinguish not just ALL from AML? That's easy. There's a stain for myeloperoxidase that lets you do that. But can we distinguish these subtypes of ALL? And so for this work, we've got, and the sample numbers aren't very high, but Eric actually managed to get this published in JPR, which I think is a testament to his ability to work statistically. Eric is actually a geneticist by training. So we've got 4 patients that have AML, 5 with ALL, 6 volunteers that are CD3 positive, and this particular stimulating factor was used, which starts them down the path to myeloid differentiation. And then we have these controls here. These cells that are purified from blood are anywhere from 60 to 90% pure. So they're not really pure. Some of them are more pure than others, but it's still a pretty good mix. And we're just doing the same thing we did with the yeast. Lyse the cells, digest the proteins, precipitate stuff that doesn't want to stay in solution, and then do quadruplicate mass spec experiments. So here, actually, this is just is coming out now in Journal of Protein Research. So what you can see is that you'll just have to trust me and read the paper if you don't trust me that Eric did all the statistical analysis correctly. The sample numbers are low, and that's what I meant by the fact that it's a testament to Eric that he was able to get this published. But I do think it's fairly tight analysis. So here's our controls, these mononucleocytes. They don't match anything other than themselves. The 3D CD34 positive cells and the AML cells are actually more closely related than these 2 cell types are to ALL. And you can see they actually look similar. If you look over here, they actually come off the same hierarchical tree. So 4 different cell types, some that are more closely related than others. And as you'll see, that's actually key to the number of proteins that we see that can differentiate between the sets. So between the sets that are the most different, CD34 positive and ALL, There's 91 proteins out of 600 roughly proteins that allow us to distinguish the two cell types. And actually, there's 10 only that all or none, Derek called it, would allow us to distinguish these cell types. And this basically means that these proteins are present in one and not present in the other cell. This is just one example of a protein that's already used that you can see, but it's already used to distinguish B cell ALS. And it wasn't detected in CD34 cells. These now are a little bit more closely related. There's 71 proteins that we can see that allow these samples to be segregated. If we go down to the two that were the most similar, there's only 17 proteins now that we can see allowed us to distinguish these two. If you take the 690, well, this is 71, so this is just AML and ALL. So you take the 71 transcripts, because Tony and Eric had already done the transcriptomic experiment for all of these same samples. You take that data and you do a hierarchical clustering of transcripts for the proteins that you could use to distinguish. And what you find out is the transcripts, while they are useful for other things, they don't allow you to distinguish the two cell types. And that's pretty much what we already know, right? Is that the transcriptomic data and the proteomic data, probably because they aren't well matched in time, collected, and the difference in lifetimes of proteins versus transcripts don't tend to correlate very well. And this is just another way to look at the fact that these don't correlate. Okay, that's basically it. So I've got 5 summaries here, and then I'll take some questions. So when we moved to Baltimore, and even before we moved to Baltimore, we're going to continue to work on this combination of digital microfluidics with SON, and probably ion mobility, because ion mobility gives us some ability to skip the chromatography step. What I'd really like to be able to do is to take that leukemia lysate, even though this is a crazy thing to suggest, I think at some point you'll be able to do it, take those cell lysates, put it on the SON chip, just scan every channel into the mass spec. So you don't have to fool with the chromatography. It becomes much simpler to read out the proteome phenotype. We're going to continue to refine the specific method. As I said, John Chapman's got it down to 24 hours. There's still some issues with this that have to do with the chromatography, I think. But we're going to continue to refine this because of the systematic nature. This may not be the best way to go about doing quantitative proteomics, but I think for building up a database, it works pretty well. And then really based on the work that we've done with Eric, we started 2 different studies, one in type 1 diabetes. It's longitudinal, where we've got samples from people over the course of 20 years from before they developed diabetes, and they went into the study as babies because their parents predisposed them to developing diabetes. And serum was basically collected every year for 20 years and went into this biobank in Finland. So we've started a study on that using Eric's approach. And then we also recently started a study of esophageal cancer, where in this case, we've got healthy and cancerous tissue from all the patients, and we're going to have genomic sequence and transcriptomic data from all the tissue. So each proteomic data set will have a genomic and a transcriptomic database to search against. So we should be much more likely to find these alternate splice events that may be controlled and regulated in certain kinds of cancers. When I moved to Baltimore, they're actually already doing multi-imaging there, and that's something we'll be getting into. And also, we've started top-down analysis, but we really haven't gotten into it with any sort of seriousness, and that also is something that we'll be getting into more seriously. And there are certain bacterial glycans that are on the surface of infectious bacteria that start to get structures up. And both of these things will happen with the new Bruker 12T ICR that I recently had to order in duress. When you get a new job, they tell you certain things, and some of those things are true and some of them aren't true. And they initially told me I had until March to spend a lot of money. And then in August, they said, well, you actually need to spend it by the end of September. So before the end of September, I had to buy or encumber the funds for 6 mass spectrometers. So that was really pretty stressful because almost certainly if you do something fast like that, I've made a mistake on one of those instruments, almost certain. Anyway, I don't want to complain because I did get the money, and I have a friend. I don't know if anybody knows Mark Emmett, but Mark Emmett moved from the Mag Lab in Tallahassee to a lab in Texas, and he got there and they said, "Sorry, we don't have any of your money." That he needed to buy his instruments. So at least they made me spend my money before I got there. That's the good news. So I want to say thanks to all of you for coming out tonight. I really do appreciate the opportunity to be here. I'm probably going to cry when I drive, lock the door, and drive away for the last time because the family's been here for 20 years almost. I should thank the funding agencies, mostly NIH and NSF, and certainly Dave Eaton, Rick Morrison, and Sam Miller all contributed money from their centers when we showed up on campus to make the lab a success. So with that, I will close and try to answer questions. That was 50 minutes. Anybody fall asleep? Yes, Jim.
What do you think the sample loss is on the sound surface?
Well, there's certainly adsorption, and there's certainly biofouling. I don't think we could really answer that unless, if we had a radio-labeled sample, that'd be the easiest way to answer that question. And it's something that we need to do, but probably not going to happen here. We could suggest Scott Edgar does that, but he probably doesn't want to work with radioactivity. I mean, there's other ways to do it. But we know that after dozens of runs, if you don't clean it, that it fouls.
But if you look at the low-intensity peptides that you see in electrospray versus SON, do you see a difference there? Just like a digest or something?
I don't know. Any of you guys want to answer that question? Did you hear the question? Well, she's down to femtomoles of peptides now. But we don't. When we started, we needed picomoles. But actually, thanks to Scott's effort and 30 minutes of slides that I cut out on technology development, they've gotten it down to a pretty sensitive level. It's not nanospray competitive, it's not atomoles, but I think we're good for many of the simple assays that you want to do. If you wanted to do an SRM after having digested something on the chip, then, I think we'd be okay with that. Dale. Well, no. I think, the samples are in acid, so there's some inherent charging from the acid that's there, just based on the pH of the compounds. It works with negative ions and positive ions. I could go down a whole list of observations, but I can't really tell you why we get these charge state distributions, except that they are usually shifted to a lower charge state, which may have something to do with electrons in the piezoelectric device, but that's just pure speculation.
Do you think this is the same as a droplet coming off the surface? Is that
No, I don't think it is. No.
That could explain the fact that you see low charge states.
Yeah.
And
Desolvation rates are different. There's a whole bunch of things that are different. There's no accelerating voltage from the time it leaves the nanospray tip to the time that it enters the heated capillary. It just sort of lumbers across. Well, this is some of the work that Scott and Yue et al. Did to improve the sensitivity. Initially, about half the droplets were about 1 micron, which is roughly regular electrospray. There were a large number of droplets that were visible like rain almost, so they were 100 microns or so. And I guess we don't know quantitatively. We should know soon but you basically have to do a measurement of droplet size, which requires a certain kind of instrument. And once those are done, we'll be able to say, since we've changed the way that we're making or nebulizing, did we actually change droplet size, which should affect rates of desolidation.
You say when you put a voltage, I assume all the protein work is done with DC voltage on the surface?
Yeah, everything ionizes better with voltage, but certainly the proteins, yeah. Well, yeah, there's probably some, like electrospray, there's some electrolytic process.
Son more forgiving for the solvent composition?
There's some experiments that I asked certain people who are here to do, but I don't think they've been done. What you'd really like to do is just put some pee on the chip or other fluids that I talked about tonight, and ask the question: do we get ions, from something that's really crude? And no, I don't think they did it. Early on, Scott Heron and I think UA put a green tea extract on the chip, and there was lots of arcing, and the front of the mass spec turned black, and they decided that they've not repeated that experiment, I don't think. Oh yeah. Well, okay. I mean, it's an obvious thing to do, right? How dirty of a sample can we use with something that's like moldy but generates electrospray charge state distributions? Okay, well, ASMS 2014 is going to be in Baltimore. So I'm hosting. Yeah, you're welcome to stop by. Hopefully we'll be up and running by then.
I want to do encore in the spring before you leave to tell us some news. Thank you very much, and good luck with your UNS.
Yeah, you bet.
And I think you know sad about leaving Seattle. You will.
Oh yeah. Well, if the company is successful, it's staying here.
Is it?
Yeah, it's actually going in the Fluke Hall in January. They've renovated it. It's an integrated. It's in a closet. Yeah.
Right.
Hi.
Nice talk. I thought I'd say hello.
Oh yeah.
You know,
Nice to
we
meet you
actually
finally.
did-